Amyotrophic Lateral Sclerosis, also known as ALS, is a disease that gradually paralyzes people because the brain is no longer able to communicate with the muscles of the body. Over time, as the muscles of the body break down, individuals living with ALS lose the ability to walk, talk, eat, swallow, and eventually breathe. The human brain remains connected to muscles through millions of specialized nerve cells called motor neurons, which help the body to move on command. The brain sends a signal along motor neurons telling a muscle to contract. In ALS, motor neurons gradually break down and die, meaning that the brain can no longer communicate with the muscles of the body. Even though the exact cause of ALS is currently being researched, ALS is widely thought to be a non-cell autonomous disease, meaning that ALS is not caused by an action within or by a single cell, but rather requires other cells to also play an important role in its development. Correspondingly, researchers found that the death of motor neurons, as seen in ALS, is brought about by contributions of several glial cells. Glia, also called glial cells, are a type of non-neuron cells found in the central nervous system, which is comprised of the brain and the spinal cord. Glial cells act as support cells in the central nervous system, and unlike neurons, they are not involved in signaling. Glial cells surround neurons and provide support for and insulation between them. Glial cells are the most abundant cell type in the central nervous system. Types of glial cells include microglia, Schwann cells, and astrocytes. Microglia are the primary immune cells of the central nervous system. They are distributed throughout the central nervous system and continuously survey the environment. They sense and react to many types of damage, such as infections, hemorrhaging of blood vessels, and abnormal protein produced by neuronal diseases. When microglia sense damage, they convert into an activated amboid or globe form to act as macrophages, clearing away debris of the dead cells. Through this process, microglia release inflammation-causing proteins called cytokines and other neurotoxic molecules, ultimately causing neuron death. Therefore, the main response to microglial action is inflammation in neurons. Microglia are beneficial up to a certain point, as they remove dysfunctional neurons. However, microglia are continuously activated by a chronic and irreversible stress brought about by ALS. ALS causes microglia to become toxic to motor neurons as they become fully activated. Schwann cells produce the myelin sheath around a neuron's axon. Myelin sheaths are sleeves of fatty tissue that protect nerve cells, allowing for a quick and accurate messaging by neurons. So far, very little is known about Schwann cell involvement in ALS pathology. Studies of human ALS show structural myelin changes along the motor neuron axons, which are most likely due to axonal degeneration. Recent studies show limited or unexpected Schwann cell involvement in the disease progression. A close relationship between Schwann cells and motor neuron axons warrants more study to help us better understand the pathology of ALS. Astrocytes are another common glial cell found in the central nervous system. Also called astroglia, these star-shaped glial cells perform many tasks in the human brain. Astrocytes provide support to the cells that comprise the blood-brain barrier, as well as maintaining the ion balance outside of cells, supplying nutrients to neurons, and aiding in post-traumatic repair and scarring processes in the brain. EEAT2 or EAT2 is a protein produced by astrocytes that plays an important role in the disease. Under normal conditions, EAT2 transports glutamate, which is a molecule that stimulates neuronal activity in the brain, back into astrocytes when communication between neurons is over, allowing neurons to rest until a new signal for activation comes in. But in ALS, the expression of the EAT2 gene is decreased, and glutamate accumulates in excessive levels around neurons, leading to their abnormal activation and damage by neuroinflammation. Regulation of EAT2 then contributes to neuronal survival and death in ALS. Changes in other cells present in the brain, such as astrocytes and microglia, accompany neuronal differences that contribute to ALS and play a crucial role in the damage and death of motor neurons. Although their activity is fundamental for neuronal health and maintenance under normal conditions, in ALS, glial cell functioning becomes damaging for the central nervous system. Further research is required to determine the exact mechanisms that cause glial cell functions to be modified